Welcome to our channel Mootechie, Structure and Functions of Prokaryotic Cell, a comprehensive visual guide to bacterial anatomy. Mootechie. In this slide, we'll explore the introduction to prokaryotic cells. Definition: includes domains bacteria and archaea, typically unicellular and small, ranging from 0.5 to 5 micrometers. This means they are microscopic life forms found in two of the three major domains of life. Key feature: they lack membrane-bound organelles. Their metabolic functions occur in the cytoplasm or at the membrane. Unlike our cells, their internal processes aren't compartmentalized within separate membrane-bound structures. Reproduction: they exhibit rapid growth via binary fission, which allows for rapid adaptability and colonization. This simple division is a key reason bacteria can spread so quickly. Ecological role: they are ubiquitous in all ecosystems and are critical for nutrient cycling, such as the carbon, nitrogen, and sulfur cycles. Essentially, they are Earth's essential recyclers. Clinical relevance: this group includes pathogens, beneficial microbiome members, and targets for antibiotics, so they are central to both disease and health. Did you know? Prokaryotic biomass on Earth is estimated to outweigh all plants and animals combined. This surprising fact highlights just how abundant these tiny organisms are. Now, let's look at the general structure of a prokaryotic cell. This diagram gives us a visual overview of the major components. Cell envelope. This refers to the complex outer layers, including the capsule, wall, and membrane that protect the cell and maintain its shape. Cytoplasm and ribosomes. This is the gel-like matrix containing enzymes and 70S ribosomes for protein synthesis. It's the cell's internal workspace. Nucleoid, DNA. This is the irregular region containing the single, circular bacterial chromosome. It's where the genetic material is stored without a surrounding nucleus. Appendages. These are external structures like flagella for movement and pili or fimbriae for attachment. Looking at the diagram, we can see labels pointing to these parts: the outer capsule, plasma membrane, cell wall, 70S ribosomes, fimbriae, DNA in the nucleoid region, cytoplasm, and a flagellum. This image effectively maps out the anatomy of a typical prokaryote. Moving on to the details of the cell envelope components. This slide compares the cell walls of gram-positive and gram-negative bacteria. Plasma membrane. This is a phospholipid bilayer with integral proteins. It's the site of respiration, ATP synthesis, and transport. Think of it as the cell's gatekeeper and power plant. Cell wall, peptidoglycan. This is a rigid mesh-like structure that maintains shape and prevents the cell from bursting due to osmotic pressure. It's like a sturdy exoskeleton. Gram-positive. Features a thick peptidoglycan layer interlaced with teichoic acids. It has no outer membrane. This thick layer holds the purple gram stain. Gram-negative. Has a thin peptidoglycan layer plus an additional outer membrane containing lipopolysaccharide or LPS and porins. This outer membrane structure causes it to stain pink. Clinical note: this cell wall structure difference determines the gram stain reaction and susceptibility to antibiotics like penicillin. This is a fundamental concept in microbiology and medicine. Key concept: the periplasmic space found between the two membranes in gram-negatives contains enzymes for nutrient breakdown. It's a specialized compartment for processing molecules. Let's dive into the cytoplasm and ribosomes, the cell's internal machinery. Cytosol matrix. This is a crowded gel-like aqueous solution, about 80% water, packed with enzymes, ions, and metabolites. It's a busy, concentrated soup where most chemistry happens. 70S ribosomes. These are composed of 50S and 30S subunits and are the primary site for protein synthesis or translation. They are smaller than the ribosomes in our own cells. Coupled processes. In prokaryotes, transcription and translation occur simultaneously, allowing for a rapid response to stimuli. There's no delay because there's no nucleus to separate these steps. Inclusions. These are storage granules like glycogen or PHB for energy, or even gas vesicles for buoyancy in some aquatic species. They are the cell's pantry or flotation devices. Cytoskeleton. Proteins like FTSZ, which forms the division ring, and MEREB, which helps maintain shape, organize the cell's internal structure. So even without complex organelles, there is internal scaffolding. The diagram shows the plasma membrane, cell wall, and inside the cytoplasm, we see many orange dots representing the 70S ribosomes, an mRNA strand being translated, an FTZ ring at the cell's midpoint, and a storage granule. This illustrates the bustling activity inside a bacterial cell. Clinical note. Bacterial 70S ribosomes are structurally distinct from eukaryotic 80S ribosomes, making them key targets for antibiotics like tetracyclines. This difference allows drugs to selectively attack bacteria without harming our cells. Now we'll examine flagella, the remarkable structures that allow bacteria to move. The diagram breaks down the bacterial flagellum structure into three main parts: the filament, which acts as the propeller, the hook, which is a flexible joint, and the basal body, which is the embedded motor. Unique architecture. It is composed of these three parts: the basal body motor, the hook universal joint, and the filament helical propeller. This assembly is a marvel of microscopic engineering. Energy source. It's powered by the proton motive force, or a sodium-ion gradient, which drives the rotary motor at incredibly high speeds. It runs on a form of electrical energy derived from the cell's metabolism. Motility patterns. Movements include run, caused by counterclockwise rotation, and tumble from clockwise rotation. This enables navigation via chemotaxis, allowing bacteria to swim towards nutrients or away from toxins. Arrangements. They come in diverse patterns: monotrichous, a single flagellum; lophotrichous, a tuft at one end; amphitrichous, flagella at both ends; and peritrichous, flagella all over the cell. Key roles. Flagella are essential for swimming motility, biofilm initiation, colonization, and they act as a specific antigen called the H antigen, which is useful for identification. In this slide, we focus on the nucleoid and genetic material. The diagram shows the nucleoid region, where the cell's single circular DNA molecule is tightly packed and condensed. Chromosome structure. It's a single circular double-stranded DNA molecule tightly packed by supercoiling and without histone proteins like in our cells. Organization. Nucleoid-associated proteins like HU and HNS organize the DNA into looped domains. It lacks a surrounding nuclear membrane, so the DNA is in direct contact with the cytoplasm. Replication. It initiates at a specific origin called ORIC and proceeds bidirectionally. This allows for rapid copying, supporting the famously fast growth rates of bacteria. Plasmids. These are small extra-chromosomal circular DNA pieces that carry non-essential genes like those for antibiotic resistance and virulence factors. They are like bonus toolkits the cell can carry. Genetic diversity. This is driven by horizontal gene transfer mechanisms, transformation, transduction, and conjugation. Bacteria can share genes with each other even across species. Key concept. Transcription and translation are coupled in prokaryotes because there is no nuclear envelope separating the DNA from the ribosomes. This allows for immediate protein production from new genetic instructions. Next, let's discuss pili and fimbriae, the structures bacteria use for attachment and interaction. The diagram illustrates different types: numerous short fimbriae for adhesion, a longer sex pillars for conjugation, and type 4 pili, which have a grappling hook mechanism. Fimbriae. Attachment pili. These are numerous, short, bristle-like fibers that enable adhesion to surfaces, tissues, and other cells. They are the primary tools for sticking to things. Sex pillars, F pillars. This is longer, thicker, and fewer in number. It forms a physical bridge between cells for DNA transfer during conjugation, a type of bacterial mating. Type 4 pili. These are unique because they can extend and retract. This grappling hook mechanism actually powers a form of surface movement called twitching motility. Biofilm formation. These structures are critical for the initial attachment and microcolony formation that leads to biofilms, aiding in long-term colonization and persistence. Pathogenicity. They act as a virulence factor by facilitating specific attachment to host tissues and helping the bacterium evade immune clearance. The key function to remember is that while flagella drive swimming, pili are the primary tools for bacterial social networking and surface interaction. This slide covers the capsule and the slime layer, the sticky outer coatings of many bacteria. The diagram, labeled bacterial glycocalyx, shows two layers, an inner capsule and a looser outer slime layer, both sitting outside the cell wall. Glycocalyx. This is a sticky gel-like coating of polysaccharides or polypeptides external to the cell wall. Think of it as a biological gel coating. Structural difference. The capsule is organized and firmly attached, while the slime layer is diffuse and unorganized. One is like a tight shell, the other like loose goo. Protective functions. This coating prevents desiccation or drying out and inhibits phagocytosis by immune cells. It's a protective shield against the environment and host defenses. Adhesion role. It facilitates attachment to surfaces like teeth or medical devices, which is the first step in initiating biofilms. As the diagram key shows, the inner gray layer represents the organized capsule and the outer light gray represents the diffuse slime layer. It's an important pathogenicity factor. Encapsulated bacteria like Streptococcus pneumonia are often more virulent because their capsule helps them evade the immune system. Finally, let's summarize the structure and functions of prokaryotic cells. Highly organized structure. Despite lacking membrane-bound organelles, prokaryotes exhibit complex internal organization optimized for efficiency. Every component has a precise role. Robust cell envelope. The cell wall, membrane, and capsule work together as a unit to protect against osmotic pressure and environmental stress. It's a multifunctional suit of armor. Rapid growth and genetics. Cytoplasmic 70S ribosomes power fast protein synthesis, while the nucleoid organizes the genome for quick replication and adaptation. Speed is a key survival strategy. External appendages. Flagella enable motility towards nutrients, while pili and fimbriae facilitate critical surface adhesion and genetic exchange through conjugation. Structure-function unity. Every anatomical feature of the prokaryotic cell is specialized to maximize survival, adaptation, and reproduction. Their simplicity is a testament to evolutionary efficiency. Thank you. And that concludes our visual guide through the incredible world of prokaryotic cells. We've seen how these remarkably efficient and adaptable organisms are built for survival. Thank you for your attention and curiosity as we explored these microscopic wonders together. Keep asking great questions.